So thank you to all of you that are here today. The plan for today is to pick up where we left off with the beginning of this chemistry content for A&P and hopefully get through a majority of the major concepts and ideas here. I will be moving through this quickly, like I was in our last meeting, because again, this should be review. So these first couple chapters going over chemistry for A&P and then cellular biology for A&P. This is all information that you should have learned in your prereqs for this course, but we're doing a refresher so that we can make sure everyone's level of understanding is at least somewhat adequate before we move on into utilizing these concepts and ideas. So where we left off last time was talking about how we can determine the overall number of each of the subatomic particles. Sorry, that word subatomic was escaping me. So how we can determine the overall number of each subatomic particle that would be present in an element using the periodic table of elements. So before we move on, I just want to use this again as a refresher. So what we have in front of us is the information that you would see on the periodic table for the element iodine. We have the atomic number of 53 and the atomic mass of 127. And so we can use this information to then determine how many protons, neutrons, and electrons would be present in the most common form of this element, the most common naturally occurring form of this element. So we're going to go through how we do this again. So would anyone be willing to share how we can determine the number of protons that would be present in one atom of iodine?
Using the atomic number in the top left.
Awesome. And so how many protons?
53.
53. Thank you, Deacon. Awesome. So as Deacon pointed out, the atomic number that comes with each element is what defines that element. So this is telling us exactly how many protons you would find in one atom of iodine. If you had any other number of protons in the element that you were looking at, that is no longer an iodine. That is definitive for this element. So iodine has 53 protons. We got that directly from the atomic number. We're also going to use this atomic number to help us with determining our number of electrons, essentially. So could someone else share how many electrons would most likely be present in one atom of iodine and why?
I would say that probably 53 as well.
Awesome. Why?
Because the number of electrons should match the number of protons. So you can have an electrically neutral atom.
Awesome, cool. That's where I was going to push you to next. Right? As these elements are represented on the periodic table, we are assuming that they are at a neutral charge or an overall charge of 0. And so if we know that there are 53 protons in one atom of this element, and each of those protons is carrying a charge of +1, so we have overall plus 53 from these protons. The number of electrons should be the same so that we can cancel out those charges with the negative charge from those electrons. So minus 53 from those electrons, giving us an overall charge of 0. Yes, James.
Are you going to have us for the exam or in any of the tests to memorize just like the amount of protons for a specific element?
No, if I were to ask you any information about a particular element, sorry, not elephant, I will either give you that information or give you a periodic table. Yeah, you do not need to memorize any of them. I suppose some of them should become second nature or should become common knowledge for you, recognizing that hydrogen is a single proton with a single electron is going to come up a lot. Oxygen being the atomic number 8 with 6 valence electrons, carbon having 4 valence electrons being the atomic number 6. So those things will come up very frequently, but you are not going to be explicitly asked to memorize that information. Okay, good question. Okay, and our last subatomic particle to identify here is how many neutrons exist in one atom of iodine. And so could someone share how we determine how many neutrons are in this atom and why?
Sure, I'll do that. So the atomic weight is the number on the bottom, or atomic mass, the 127 or 126.9, and you subtract the number of protons, 53, from the atomic weight, and then you get the number of neutrons.
Awesome. So 74 in this case, right? If we do that subtraction. So the only subatomic particles in this, or in any element that carry mass, are going to be the protons and neutrons. And so if we've already determined the number of protons from our atomic number, we can then just subtract that number from our atomic mass, and we know that there must be 74 neutrons left to give us that total mass, right? So protons and neutrons both carry a mass, and they are located in the nucleus, or the center of the element or of the atom. Electrons do not carry a mass. However, they carry a charge of -1. Protons carry a charge of +1, and neutrons do not contribute to the charge of the molecule. Okay. Cool. So great job, and thank you guys for going through that review. So electrons are what we're going to be spending most of our time talking about. Because the electrons in an atom are what are going to determine and be utilized in order to make bonds with other atoms or with other elements. And so we refer to the location of these electrons as clouds or shells. You could use either of those words, so an electron cloud or an electron shell. And essentially, each of these shells is representative of an energy level that those electrons would be carrying or are participating in. And so each of those shells also has a maximum number of electrons that it can hold. So the first shell, which is going to be the closest to the nucleus. Okay, so here we're only showing the nucleus with a single electron shell or electron cloud. This first shell can only hold a maximum of 2 electrons. Moving beyond that into into shells that are farther from the nucleus, they can hold more and more electrons. And so the second shell can hold up to 8. The third shell can hold up to 8. When we get into much larger elements, that rule and that number gets a little bit variable. It's not always an exact 8. But for the purposes of this course, for the purpose of most biological molecules, we are only really caring about these first 3 shells. Okay. Another important concept here is that electrons will always exist on the shell closest to the nucleus first. So if we're looking at the elements like lithium here, lithium has the atomic number 3. And so as we just practiced, that means it has 3 protons, which means it also has 3 electrons. One atom of lithium, where is the whiteboard? They've moved the whiteboard button on here. I can't draw. Okay, well, the point was going to be lithium's 3 electrons would be located first in the first shell. So the first 2 would fill up that shell, and then the third electron would then be on the second shell. And so the closest shells to the nucleus are always going to fill up first. Okay, cool. And so whichever shells are left over on the outer shell, on the outermost shell, are called the valence electrons. And atoms love to have full valence shells. So since the outermost electrons are called valence electrons, we also refer to that outermost shell as the valence shell. And that is what is going to be most important when looking at how these atoms, how these elements are making bonds with each other to form molecules. Okay. And again, typically the number we're going to be looking at as that maximum full valence shell is going to be 8. The only exception, as far as what we are concerned with, is hydrogen. Since hydrogen only has 1 electron, it only has 1 electron shell. And that is the first shell, which can only hold a maximum of 2. Okay? So that first shell only holds 2 electrons. So hydrogen is perfectly happy with a full valence shell of 2 electrons. Every other element is going to be reaching for 8 total. Okay, so moving on from that, there are elements that already have full valence shells. So any of these elements that exist in group 18, so helium, neon, argon, etc. any elements in group 18 here has a full valence shell on their own, and so they cannot create chemical bonds with other elements. They only exist naturally or can create with themselves. They may have interactions with other molecules or other elements, but they cannot form full bonds. They cannot share electrons with each other. Okay. And so a bond, that term bond, is referring to any time two or more atoms are sharing or exchanging those valence electrons. And so depending on which action here is happening, that is what is going to determine what type of bond we're looking at. So again, atoms can either share their electrons or they can exchange electrons. Cool. So here's a look at what some of these smaller elements look like with their valence electron shells or with their electrons placed into shells. So hydrogen only has that first shell. It has one electron in that first shell, so it is able to make one bond, essentially. Helium, neon, and argon are all part of group 18. They have full valence shells and cannot bond with any of them. But some of the more important ones that we'll be talking about a lot are carbon and oxygen. So carbon is the atomic number 6. So it will have 6 electrons. The first 2 fill up that first shell. And so we're only left with 4 valence electrons on a carbon. And that puts carbon in a unique position where it has the highest number of available bonds compared to these other elements listed here. And it has 4 valence electrons, therefore it has 4 possible bonds that it could make with other elements. Same goes for silicon. But carbon is what we'll be seeing show up most in living organisms. Carbon is the backbone of all organic molecules that make up living organisms, including humans, right? And then oxygen here, which we'll be talking a lot about in this PowerPoint, has 6 valence electrons. And so it only has 2 available bonds, but because it has almost a full valence shell already, it is very electronegative. So a new term there, electronegative, meaning that it has a very strong pull on shared electrons. So whenever oxygen is bonding with other elements, it typically is going to have a stronger pull on any shared electrons, which is going to give it a partial charge, which we'll talk about more here in a second. So oxygen is very electronegative. It has a strong pull on any shared electrons. Okay, so now moving into bond types. Shown here is a nonpolar covalent bond. So covalent bonds are any bonds where electrons are being shared. And so typically if one element is bonding with another atom of the same element, they are going to have an equal pull on the electrons they are sharing. And so they are sharing, making it covalent. And because the pull on those electrons is equal, it is non-polar. And that may make more sense when we talk about polar bonds, polar covalent bonds. So again, covalent bonds can either be polar or non-polar. Both have shared electrons, but it is the pull on those shared electrons that will determine what type of covalent bond. So water is going to be our main example of a polar covalently bonded molecule. Oxygen bonded with 2 hydrogens here. So each of the hydrogens is sharing its 1 electron with oxygen. Oxygen now has 8 valence electrons, so it is happy. Each of these hydrogens now have 2 valence electrons, so they are happy. However, as we just discussed, oxygen is very electronegative, especially compared to hydrogen. And so although these electrons are being shared, they actually spend more time closer to the oxygen than they do to the hydrogen, leaving the oxygen with a slightly negative charge and each of the hydrogens with a slightly positive charge. And so that's what gives us our poles, making it polar. I really wish I had a whiteboard. I'm gonna have to figure that out in my Zoom settings as to why I don't have a whiteboard showing up. But essentially what I would draw here is just to indicate this oxygen you should consider with a negative charge on this side of the molecule, and these hydrogens both creating a slight positive charge on this side of the molecule. So it is essentially now a magnet, right? It has both a positive and a negative end. And so this means any other molecule or atom that is nearby a molecule of water and is carrying some sort of charge, water is going to be able to interact with it in some way, some magnetic pull, right? right? That idea of opposites attract. Anytime negative charges and positive charges come near each other, they will automatically form an attraction, a pull. And that is what gives water most of its properties that make it so critical for life. So the oxygen side of the molecule will form attractions with any positively charged portion of a molecule or ion, and then the hydrogens will be able to form attractions with any negatively charged portion of a molecule or ion. And so that forms a polar covalent bond. We get two poles. We get a negative pole and a positive pole. Okay. Carbon typically creates nonpolar covalent bonds. So carbon is not very electronegative. It is very generous with its sharing of electrons. And so almost every element that carbon bonds with, it is going to form a nonpolar covalent bond. The exception comes from when we have very electronegative elements bonding with that carbon. So like the oxygen would have a stronger pull on the shared electrons if it bonded with carbon. Okay. So is what I'm saying so far making sense? Is there anything you guys would like to ask about or clarify?
And it's like a weak what is it, polar charge with water, right? That's kind of what gives it its like stickiness. So it like will adhere to things, but not all the way or something like that.
Yeah, so the polar nature of this molecule, the fact that it now is carrying these slight charges, allows it to interact with other molecules that have opposite charges. And that pole is a weak attraction that we refer to as a hydrogen bond very commonly. Right? And so that is where we were going next. So that's the perfect question right there. So like I said, that magnetic pull is going to create some slight attraction. So if another polar molecule like ammonia, which is nitrogen is also very electronegative. So if ammonia is to come near a molecule of water, the slightly negative pole of the nitrogen will form an attraction with the slightly positive pole of hydrogen here, and vice versa, right? The hydrogens in this ammonia could form a hydrogen bond with the oxygen from water. And so these kind of magnetic attractions between polar molecules are called hydrogen bonds. Oops. Okay. So a hydrogen bond is not a bond in the same way that we were discussing before. I wish they had given it a different term, but this is how it's talked about. Because there is no sharing of electrons or exchanging of electrons happening here. They are simply being drawn to each other by that magnetic attraction. And as a result of that, water gets all those characteristics, such as what James brought up of kind of the stickiness of how water can hold on to other water molecules. Okay? And they are constantly breaking and reforming these hydrogen bonds over and over. Right? And so for the purposes of this course, you don't need some of the characteristics of water that are discussed on these few slides here, but please feel free to take a look at it, such as whatever, surface tension, cohesion, adhesion, the whole reason that ice floats, right? These are all important things, as well as how water deals with changes in temperature. These are all important things for supporting life. But as far as what we need in this course, we're going to move on to the way it affects solutions, or the way it's used in solutions. So because water has this ability to interact with other things, it is a polar molecule, and because water is so prevalent in all environments and in all living things, right? So the inside of almost all of your cells is has a very high concentration of water. It is full of an aqueous solution, and almost all cells are surrounded by some sort of aqueous solution. And so we can define different substances or molecules, right, solutes, that exist in environments based on how they interact with water. So we could define a substance as either hydrophilic or hydrophobic. And if we break that word apart, hydro referring to water, phile or philic meaning a love of, and then phobe or phobic meaning a fear of. And so any hydrophilic substance or molecule is one that can bond with water, can interact with water. So if water came near it, it would form that hydrogen bond. Okay, so hydrophilic substances can bond with or interact with water. Hydrophobic substances cannot. So any molecule that is not carrying a charge is not going to be able to form those hydrogen bonds or interactions with water. And that plays a big role in the structure of the phospholipid bilayer, which makes up the cell membranes of all cells. So all the cells in your body, all cells of living things, have a phospholipid bilayer. And portions of these phospholipids are both hydrophilic and hydrophobic, and thus this controls or gives some control as to what is crossing this barrier. And we'll go into more depth in a few slides here about this. But so we describe substances as being either hydrophilic or hydrophobic based on how they interact with water. And that can give you a lot of insight into how that substance will interact in a living organism in general as well. All right, so here's a closer look at that phospholipid bilayer. Right? So again, this is a cell membrane that would be completely surrounding a cell. And the internal environment of a cell is going to be aqueous or water-based. The external environment of almost all cells is going to be aqueous or water-based. And so the structure of these phospholipids is kind of set up in a way that that allows it to interact with water while also preventing everything from just freely moving in and out of the cell. So a phospholipid is considered amphipathic. This is our last term related to how molecules interact with water. Amphipathic, meaning that it has both a hydrophobic and a hydrophilic region. So the phospholipid itself is composed of a glycerol head with a phosphate attached to it. And this glycerol head is hydrophilic. It can interact with water. And then these phospholipids also have two fatty acid tails. And these fatty acid tails are nonpolar, meaning they are hydrophobic, and so they cannot interact with water. And so this membrane that surrounds your cells has the hydrophilic heads pointing outwards and inwards. And then the inner portion of the membrane is completely blocked off, essentially, by these hydrophobic tails. Right? And so that's going to play a big role in what can and cannot cross this membrane, which, like I said, we'll talk more about in the next chapter. And I think there's even a couple more slides in this slide deck. Okay. So as far as chemical bonds, so far we talked about our nonpolar covalent bonds, which does not produce a charge, but they are sharing their electrons. We talked about polar covalent bonds, where they are sharing electrons. However, one element is going to have a stronger pull on those electrons than the other, which gives the overall molecule a charge, making it polar. We talked about hydrogen bonds that form as a result of polar molecules. And then lastly, we're going to talk about ionic bonds. So an ionic bond is, what's the word I'm looking for? It's escaping me. But an ionic bond is significantly different from a covalent bond, definitively different than a covalent bond, because there is no sharing of electrons forming the bond here. Instead, there is an exchange of electrons. So ionic bonds are most common between elements that only have one valence electron when they are on their own, and elements that have 7 valence electrons. Okay? So since this chlorine here has 7 valence electrons, it only needs 1 more electron. If sodium were to share this lone electron, that would satisfy the chlorine. Right? Chlorine would get 1 more, so now has 8. But then sodium is just stuck with 2 valence electrons, never to have a full shell. It will never be able to fill the rest of that shell. So instead, rather than trying to fill the outer shell that it has, it gives away this electron. Chlorine essentially steals it. And now this outer valence shell disappears, and it goes down to its full second shell. That's what we have shown here. So when sodium and chlorine come into close contact with each other, sodium will give away that valence electron to chlorine. It then loses that outer shell and now has a full valence shell left behind. And chlorine now has a full valence shell. Okay. But since we have exchanged electrons, sodium now has 1 less electron than it does proton, right? It started with 11 protons and 11 electrons. It gave away 1 of its electrons. So now it has 11 protons, but only 10 electrons, leaving it with an overall charge of +1. is this font like big enough for you guys to see on your screen? Like on my screen, it's large, but I don't know what it looks like for you all. Is this an effective way to write things?
Yeah.
At the
Okay.
bottom left.
Yeah, this specifically as I'm typing.
Oh, I didn't actually see it. It was kind of the little window screen was covering it. So I see it now. It's fine. It's not super big. I notice it now, so it's fine.
I'm gonna get the whiteboard thing figured out, and that will hopefully be larger. So anyways, since sodium started with 11 protons and 11 electrons, it gave away its electron, so it now has one more proton than it does electron, and so it has one more positive charge than it does negative. So sodium in this bond has now become a positive ion. So an ion is any element that is carrying a charge. Sodium gave away an electron and now has a positive charge. A positively charged ion is referred to as a cation. So cat with a T, you could think of that T as a plus. Cations, T is a plus, have a positive charge. The chlorine essentially had the opposite happen to it. It started with, 17 protons and 17 electrons. It has now gained an extra electron, so it has 18, leaving it with an overall charge of -1. And so a negatively charged ion is called an anion. So if you can at least remember that these two terms, cation and anion, are referring to ions, and if you can remember, look for that T, which looks like a plus sign, right? Cations are positively charged, anions are negatively charged. Okay? And so we still have not formed a bond yet. Okay, we've had an exchange of electrons based on how electronegative chlorine is. It just straight up steals that electron. But the bond comes from the fact that they have now gained these new charges. So sodium now being a cation and chlorine now being a negative or an anion, right? They can now form a stronger magnetic attraction than what we saw with water, which is what creates our ionic bond. So it's an ionic bond because we made ions. Those ions now attract to each other like a strong magnet, and that will bond them together, at least temporarily. Did you have a question, James? I saw that hand for a sec.
No, I didn't want to. It's kind of about the last one. I always forget polar versus non-polar. I was wondering if there was a way that's like an easy kind of mnemonic or whatever way to say, oh, this one they're sharing them equally or not. Between
Well, the term polar, right, referring to poles. So if you can get that stuck in there, like the poles have their own unique charges in the same way that we have a north and a south pole that have different chemical charges to them. And that's how the compass works. Polar molecules have two poles to them with a separate charge. They have distinct sides. Whereas a non-polar molecule would not have that same distinction. It does not carry a charge.
I think that actually does make sense. Thank you for that. Yeah.
Yeah, polar for poles. Okay. And so ionic bonds are typically what form what we call salts. So this is bonding between minerals, right, or micronutrients in our body. These are minerals. And these ionically bonded salts will typically form large salt complexes, such as this, a crystal, right? And so this is what you're familiar with seeing in like your table salts that you would use with your food, right? It has those larger crystals of salt. That's just a bunch of teeny tiny sodium and chlorine ions. And then as soon as that crystal is added to water or any aqueous solution, it will dissolve. And it's not disappearing. Those sodium and chlorine ions are still present in that solution. They're just being separated from each other. That ionic bond is being interrupted by the polar water molecules that are present in the cup of water you just added it to. Okay? So because water is polar, it can form those slight interactions with these ions as well, essentially just pulling them apart. Okay? So the positively charged sodium is going to form interactions with the partially negatively charged oxygens on water. And so those water molecules will surround the sodium. At the same time, the slightly positively charged hydrogens are going to surround the chlorine, and that just kind of pulls them apart from each other. And that is what dissolving is in a nutshell, right? Water creates these hydration spheres, okay, surrounding ions and pulling them away from each other. And it can do a similar thing with polar molecules, right, such as crystals of sugar. Each of those glucose molecules in that sugar is slightly polar or has polar regions to it. And so the water can separate glucose from other glucose molecules by creating these hydration spheres in the same way. So here's a closer look at those hydration spheres themselves. So if this were a full aqueous solution, as soon as you add the salt to it, water will immediately start to form these bonds, creating hydration spheres, separating these ions from each other. And again, can do the same thing to molecules that have polar regions or are polar themselves. Okay. And so these minerals, these ions that we get from our food mainly, right, or essentially everything, all the nutrients, all the molecules that you're taking into your body are going to have to come from your food. Right? A lot of the minerals and ions are going to be used throughout the cell for things like cellular communication and movement of molecules in and out of the cell itself. So we're going to be talking a lot about which ions are naturally in highest concentrations, either outside of the cell or inside of the cell. We'll look at examples of how the cell moves these ions and what results that produces, such as the movement of sodium and potassium to generate an electrochemical potential, which is what your neurons use to send signals to each other, things like that. Okay. Let's see, is there anything that we haven't already discussed that's important here? So electrolytes are another fancy term for salts. Essentially, electrolytes are any soluble inorganic molecule. So that's going to be mainly minerals. If it's inorganic, it is not based in carbon. It does not have a carbon-based backbone. And so again, those are just different types of salts. So sodium chloride, potassium chloride, calcium phosphate, these are all technically salts, also called electrolytes. Okay. So the last thing on just water here is how water participates in larger chemical reactions. Right? So we've looked at how water can form these hydrogen bonds, these interactions with molecules, how water can form hydration spheres to help dissolve molecules. But water is also the basis for most of the building and breaking apart of any large organic molecule. So organic molecules have a backbone of carbon and are typically relatively high in oxygen and hydrogen as well, or these are common elements that would be bonded with those carbons. And so we can form bonds between organic molecules by removing a water, which is what you see here. So this hydroxyl group that was originally attached to this carbon and this hydrogen from the other hydroxyl group are going to be removed. That frees up a valence electron on this carbon and frees up a valence electron on this oxygen. And that allows them to now bond if we add some energy to this system. So we've taken out a water molecule, we've added some energy to create this new bond. And now the energy is stored in that bond. And so the example you're seeing here is two different monosaccharides. These are both simple sugars, a glucose and a fructose. We've removed water to create a new bond. And now this is a disaccharide made up of two sugars that we call sucrose. And we got water as a result of building that bond. So most, I'm not confident enough to say all, but most building of organic molecules, of macromolecules in our body is going to occur through dehydration synthesis. So dehydration meaning we're removing a water from those two molecules in order to create a new bond. And so in the same way, the opposite is true. So hydrolysis is referring to the addition of water to some large molecule in order to break that bond, right? And now we have added back that same hydroxyl group we were looking at earlier and that same hydrogen onto this hydroxyl group. So by adding water, we can break bonds. By removing water, we can build bonds. Of course, that also requires energy on both sides, but that's a general idea. Okay. So again, gonna make an explicit attempt to ask for any questions if you guys have them. Feeling okay with water? typically in a face-to-face classroom, I would do like a fist to 5 thing to get an idea. I don't know how to, or I'll have to come up with a way to do the same thing here. But the idea would be, I would ask everyone to show with their hand. A fist is a 0, which I have no idea what's going on here. A 1 would be like I have some idea, but still very lost. And then a 5 is I completely get it. I have a good understanding. 4 is close to that, right? So we could try that a little bit, at least with those that have their camera on. Can I get a fist to 5 on how you're feeling about chemical bonds in water? Threes. Okay, a good amount of 5s and some 3s and 4s. Okay, so thank you. That's good to know. And so again, if there's any part of this that feels iffy or the way I described it didn't really make sense, please give me the opportunity to clarify or to make up for that by asking a question.
Excuse me, sir. I had previously encountered this, and instead of dehydration synthesis, we referred to it as condensation. Would that
Okay.
still apply here?
I think I have heard that term before as well. To refer to this, I think most commonly now it's dehydration synthesis. Almost all biology textbooks that I've seen, or textbooks that relate to biology, are now using dehydration synthesis very commonly. But if you do come across a resource that refers to a chemical reaction as you said, condensation, that is likely referring to the same idea. Right? It is creating a new water molecule by removing it from these other reactants. Okay, so now getting into pH and buffers. This, as a concept that applies to biology itself, was one of the things that took me the longest, I think, to actually fully develop a deep understanding of, or to feel like I understood it, really. For a long time, I would just have to check notes or look up stuff to understand if I'm looking at an acid or a base. So I recognize this is somewhat complex, but essentially, at the most basic level, pH is referring to or reliant on the concentration of hydrogen ions. Right? So this H, right? So it's referring to the concentration of hydrogen ions that are present in a solution, specifically hydrogen cation ions, which would be a single proton. And so to exemplify that point before I move on, I think I had a slide showing hydrogen here, right? Hydrogen is the atomic number 1. Hydrogen is an atomic mass of 1. So let me actually go to the periodic table. So an atomic number 1, an atomic mass of 1. So that means hydrogen has 1 proton, 1 electron, and 0 neutrons. Okay. And so hydrogen ions can either be a proton themselves or an electron themselves. So these hydrogen ions be 1 proton, which would be H+, or 1 electron, be H-, right? So if you see a hydrogen ion being represented that way, they are referring to either a proton or an electron. In the case of pH, we care about the protons, or the concentration of protons is what determines the pH of a solution, where a very low pH is referred to as an acid. So if there are a lot of these free protons in the solution, it is going to have a very low pH, and it is going to be very acidic. So the more free protons, the more acidic that solution is. And so for a base with a very high pH level, there are not very many free protons, or there's a low concentration of free protons. You could also look at it as having a high concentration of these hydroxides, which would take up those free protons. But I think the simplest way to look at it, or the way that I want you guys to think about this, is any acid is going to be a solution that freely gives away protons. Any base is going to be a solution that takes away protons. Right? So either turns it into water or makes a bond with it in some other portion of the molecule. So acids give away protons, bases take protons. And I think with that level of definition, at least for most biology courses, that is going to apply and that is going to be true. If you're going into chemistry or deeper chemistry, it's going to get into a lot more of the specifics about it. But for the purposes of this course, again, acids give away free protons, bases take free protons. So they've removed them from the system. And so you would calculate, the pH of any solution by taking the negative log. Oh, I thought that was in here, and that's why I started saying it. By taking the negative log of the concentration of protons. That's kind of what's described down here. So the measurement for it, I'm just going to put it actually in the slide. So to calculate pH, you take the negative logarithm of the concentration of free protons. So anytime you see these brackets in chemistry, this is referring to concentration. So the negative log of the concentration of free protons. Okay. I don't expect to ask you to do this. It's just plugging in numbers into a calculator and using the logarithm thing. But it's good to be aware of. Other classes might ask you to know how to calculate the pH. And this is just another visualization of the same idea. So a very low pH, with the lowest being 0, all the way up to a very high pH with the highest being 14 is the entire pH scale. So the lowest or the most acidic solution would be a 0 or 1, and the most basic solution would be 14. And so in the center here, a neutral solution with a pH of 7 would not have an overwhelming free concentration of protons or an overwhelming concentration of these hydroxide groups, which steal protons. And so that is a neutral solution. And water is our perfect neutral of 7. If you split water apart, you can take one proton off of a water molecule, and that would give you this H+, and it will leave behind this hydroxide group. Right? So water is basically an acid and a base together. It is the perfect buffer to help reduce the acidity or reduce the basicness of a solution. Oh, cool. I forgot I added this slide in here. Awesome. So this is what I was looking for here. So you should consider any acid or an acid as any solute that dissociates and releases hydrogen ions, so releases those protons, thereby lowering the pH. And you should consider any base or a base as any solute that removes those protons from the solution, thus increasing the pH. A buffer is then a compound that would stabilize the pH of a solution. So essentially moving it closer to 7, that neutral 7. So a buffer is any compound that is going to change the pH of a solution, bringing it closer to 7. Okay, and so the last portion of this, chapter is now going into the major molecules that make up a majority of the mass of living things. So these are our biomolecules or macronutrients, macromolecules, if you have that nutrition background. And so we have 4 categories of molecules that make up a majority of the mass in a living thing. These are our carbohydrates, aka sugars, lipids, aka fats, proteins, and then nucleic acids. All of these are organic molecules, meaning they have a carbon backbone. And then it is just a difference in what other elements and functional groups bond with that carbon backbone that end up giving them their unique characteristics. So for carbohydrates, we're going to be looking at the different forms of carbohydrates that are important to the human body. So a single carbohydrate is a monosaccharide. Mono is a prefix meaning 1. Right? We have di as a prefix meaning 2, poly as a prefix meaning many, and then saccharide or saccharide is the Latin root for sugar. So a monosaccharide is a single sugar, a single molecule of a carbohydrate. Disaccharide is 2, and a poly is 3 or more. For lipids, we have a few different types of lipids. Again, this is another word for our fats that exist in our body. So we'll be talking mainly about fatty acids, steroids, and phospholipids, a little bit about glycolipids in this class. Proteins are long chains of amino acids. So the monomer or the building block of a protein is an amino acid. And those chains of amino acids end up forming interactions with each other or get folded by another protein into a specific shape that allows them to perform whatever function they are responsible for. And then lastly, our nucleic acids are what make up all of our genetic material, so DNA and RNA. And essentially, all that nucleic acids do, all that DNA and RNA are, is instructions for producing proteins. So all of your genetic information, everything you inherited from your mother and father, right, were simply instructions for producing proteins. Those proteins are then what determine the physical expression of that gene. Right? And by physical, I don't just mean like what we can sense visually, like hair color, eye color, skin color. That is a part of this. Proteins determine that, but also what your cells are capable of. So what enzymes they produce, and thus what chemical reactions can occur, and at what and how often and at what rate, such as the ability to digest lactose. In order to digest lactose, you need to have the gene that codes for the protein lactase. And so if you don't have that gene, you don't make that protein, and now your body cannot digest lactose. Right? Okay, so we're going to dive into all four categories here, talking about the major elements and at what ratios those elements show up in these molecules, as well as looking at their functional groups. So again, each of these biomolecules has a carbon backbone, so mainly carbon as the center element holding these groups together, but they differ in their functional groups. So what other elements are bonded to that carbon backbone? And that is what gives them their specific functions. Right? So amino acids have an amino group, that's the name, that's what makes up proteins. Both fatty acids and amino acids have a carboxyl group. Carbohydrates, fatty acids, and amino acids all have at least one hydroxyl group. And then phospholipids, nucleic acids, and ATP, which is our last like biomolecule here that we'll talk about, all have phosphate groups. So phosphates are really great for energy, as well as adding a polar portion to a molecule. So first with carbohydrates. Carbohydrates are the simplest of our biological molecules here. They always have this carbon to hydrogen to oxygen ratio of 1:1. Almost all monosaccharides, so single sugars, are going to have 6 carbons. And thus, if we follow this ratio, almost all monosaccharides should be C6H12O6. Sorry that I don't have the subscript there working quickly. No, come back. Okay, hopefully you guys understand. These are just subscripts, right? So in this one molecule, there are 6 carbons, 12 hydrogens, and 6 oxygens, right? So that ratio of 1 to 2 to 1. And that should be true for almost all monosaccharides, right? And so these carbohydrates can be linear. A lot of the time they form these ring structures. So as we move on and look at the chemical structure of some of these other biomolecules, you will see this ring as a part of their structure. And that is a carbohydrate that makes up a small portion of another biomolecule. Right? So again, the important part here is this ratio of carbons to hydrogens and oxygens, and the fact that they can form these rings, these ring structures. And so essentially each of the bonds holding these carbons together is storing some potential energy that can be released if we break that bond. And that is the basis of cellular metabolism, right? Of cellular respiration. Right? You start with a glucose molecule, we break the bonds holding these carbons together. Every time we break that bond, some energy gets released, and eventually we transfer that energy into ATP, which can then be used for the entire cell. So the most common polysaccharide that we're going to be talking about in this class is glycogen. Glycogen is the storage form of carbohydrate in animals. Right? So plants have starch. Right? A potato is a storage organ for starch in plants, which essentially looks like the same as this molecule that we're looking at here as glycogen. But in animals, we make glycogen. And so glycogen is just a whole bunch of these glucose molecules bonded together. And thus, we are storing glucose for when we need it, mainly in the liver, as well as a lot of our skeletal muscles, where it's in high concentrations in our skeletal muscles. So the parts of our body that are taking in food, like the liver, carbohydrates go to the liver first after you digest them. And then the parts of our body that need that energy most, so the skeletal muscles. Carbohydrates are also the only fuel that can be used, or I don't know if I'm phrasing that well. So let me put it a different way. Red blood cells and neurons rely extremely heavily on glucose, on carbohydrates as their energy source. They cannot use anything else. So carbohydrates particularly important for our red blood cells and for our neurons. And so in animals, the most common monosaccharides you would come across are glucose and fructose. So again, these are both simple sugars. They are a single molecule of 6 carbons. The most common disaccharides are sucrose, lactose, and maltose. And then the most common polysaccharide, glycogen. That's that storage of glucose. So moving on to lipids, very similar to carbohydrates in that we still only have carbon, hydrogen, and oxygen making up these molecules, with the exception of phospholipids, which we'll get to in a second. But lipids are also commonly referred to as fats. They have a carbon to hydrogen ratio of 1 to 2, the same way that carbohydrates did. So our carbon to hydrogen ratio is 1 to 2. However, lipids have a much lower number of oxygen. Okay? And the main portion or the largest portion of each lipid is going to be its fatty acid tail. So essentially the monomers, the building blocks of lipids are fatty acid tails and their glycerol heads, which I'm going to skip ahead to a little bit here. So here we're looking at a phospholipid, but I just want to point you to the glycerol head here. So we have 3 carbons. This is technically a small sugar, a small carbohydrate. So we have a glycerol head and fatty acid tails. This is going to make up the core of every lipid, glycerol head with fatty acid tails. But again, the largest portion are these fatty acid tails themselves, which are just long chains of carbon with hydrogens off of them. And so fatty acids can either be considered saturated or unsaturated, and each of them comes with unique properties. So saturated is referring to the saturation of each carbon with hydrogens. So right here, we're looking at a saturated fatty acid tail. Every single carbon in this fatty acid chain has 1 bond going to the next carbon and 1 bond with the previous one. And the rest of its available bonds are saturated by hydrogen. Comparing this to an unsaturated fatty acid, which would have at least 1 double bond between carbons. So thus removing the available number of bonds for hydrogen, essentially. So I think it's easiest to just think about unsaturated or simply focus on this double bond when keeping these 2 terms straight. So saturated is fully linear, only single bonds between each carbon. Unsaturated fatty acids have at least 1 double bond. And that double bond creates what we refer to as a kink. A kink, which is this fold you see shown in the 3-dimensional diagram. So a saturated fatty acid is completely linear almost. An unsaturated fatty acid with this double bond is going to create a bend or a kink. And what this does is it creates space between neighboring fatty acids. I really wish I could draw here because I don't think I have any good unsaturated fats here. I guess this is a decent look at it. So just looking at the tails here, we have a saturated fatty acid tail and an unsaturated fatty acid tail. That double bond creates that kink, and now it's created a whole bunch of space that wasn't there before. So by creating this bend, it is now creating space between neighboring fatty acids. And because there is more space, that allows this lipid to be more liquid. It is more fluid. There's more space to move around, and thus it is more likely to be liquid at room temperature or at our typical body temperature of 98.6 degrees Fahrenheit, right? And so that's the main difference. Unsaturated fatty acids are more likely to be liquid because of the double bond creating a kink. So oils or any other fat that is liquid at room temperature, those are all mainly unsaturated fatty acids. Whereas saturated fatty acids would be those foods and fats that are solid at room temperature, like butter and lard. Okay. So now looking at the major versions of lipids. So we'll mostly be talking about triglycerides, steroids or sterols, phospholipids, and then glycolipids. But before I get into this, yes, James.
And what makes them harder or easier for our body to like break down, or what makes them more dangerous with regards to fats? Like I
Yeah.
remember like trans fats, or even where is the way they stack or something.
Yeah, so a trans fat is an unsaturated fatty acid with their one hydrogen bond being on the same side. I guess I'm trying to think of like what level to go into this here as far as what's important for this class. Let's go further into that in lab as far as looking at the trans fats and why a trans fat might be less healthy than a cis fat. But I do want to address, it's very common historically and even still today, like government agencies that produce like uh, diet plans and determine what is healthy and share that with the public are still saying that saturated fat is unhealthy for you. However, most of our modern research and review shows that that is not true, or at least is not strongly supported. Saturated fat should be perfectly fine for your body. Your cells use it just fine. And then, of course, unsaturated fats, which we have always considered to be healthy and are the easiest for your body to use. But both saturated and unsaturated are fine. It's just the trans fats you should avoid. And we can dive further into that in lab. Just want to make sure we get through this. Okay. So the major dietary fat, the major lipid that you are getting from your food is going to come in the form of triglycerides. So a triglyceride, referring to the fact that we have 3 fatty acids here, attached to one glycerol head. So this is a fully nonpolar molecule. And we have a carbohydrate here, a small carbohydrate with this glycerol head, and we have 3 fatty acid tails. Those fatty acids can be added to the glycerol head through dehydration, aka condensation synthesis, or they can be disattached or removed from the glycerol head through hydrolysis, through adding water to it. Okay. So you could also refer to a glycerol head with 2 fatty acids as a diglyceride, or a glycerol head with 1 fatty acid as a monoglyceride. So next are steroids. So these are larger lipids that can travel through the body as hormones in some cases, like estrogen and testosterone. Those are both lipids that act as hormones, so long-range chemical messengers. The bile salts that we produce and put into our small intestine to help digest fats, those are steroids or sterols. And then cholesterol, a very, very common sterol or lipid in our bodies, is very helpful for adding some stability and rigidity to our plasma membranes. So our plasma membranes are made up of these phospholipids, right? We just looked at the bilayer. I can pull one up again here. Right? So this is what a typical membrane looks like. We have 2 layers of these phospholipids. Phospholipids are liquid at room temperature. Or sorry, not at room temperature, but at our body temperature at 98.6 degrees Fahrenheit, they are liquid. And that's not always advantageous, or it could be dangerous as far as keeping that membrane together. And so cholesterol, these sterols here, kind of inject themselves to provide stability. And so our membranes or our cells can move cholesterol throughout the membrane to provide more stability in one area versus another. We can add more cholesterol to provide more rigidity and stability to the membrane. Right? But cholesterol is mainly used in that way to add some solidity or to help solidify any other lipids that would make up a membrane. So not just the cell membrane, but also the membranes that surround all of your organelles. And so a phospholipid is unique to the other types we've talked about so far because it carries a phosphate group. So it still has a glycerol head. In this case, each phospholipid only has 2 fatty acid tails, but it also carries this phosphate group, which provides a polar region to this molecule. And so that makes a lipid that not only has its nonpolar tails, these hydrophobic nonpolar tails, but now also has a polar hydrophobic head. And so these heads can interact with water. They aren't being repelled or pushed away by water in the same way the tails are. And so that's what makes them great for our plasma membranes. Again, those heads are pointing inwards to the aqueous environment inside the cell and outwards to the aqueous environment surrounding the cell. And then the nonpolar hydrophobic tails are pushed in towards the center, preventing the free movement or diffusion of a lot of molecules, right? Giving some selective permeability, some control over what is just entering and leaving the cell. Right. The last main type of lipid to talk about here, right? Yeah, is just our glycolipids. So if you break this word apart, glyco, okay, we've been looking at glucose and glycogen, so referring to some type of carbohydrate, and lipid. So this is a carbohydrate group attached to our glycerol head of a lipid. And so these carbohydrates would be sticking out of the plasma membrane of a cell. And this is what is used for cells to recognize each other, essentially, as neighbors, or to form an extracellular matrix that holds two cells together, like in a tissue, okay? Through these carbohydrates that are poking out of the membrane. Okay? So this is our glycolipid here, and you can see those carbohydrates sticking out, those ring structures, right, with the fatty acid tails embedded into the membrane, essentially. All right. We're going to talk a lot more about cell membrane in the next chapter too, and I've kind of said all this stuff over and over again, so we will come back to that later. So anything you guys would like me to touch back on about carbohydrates or lipids? Okay. So again, so far carbohydrates have that carbon, hydrogen, oxygen ratio of 1 to 2 to 1. Lipids have a carbon to hydrogen ratio of 1 to 2, but typically have much less oxygen. And now moving into proteins, which are made up of amino acids, we're going to start getting some other elements into the mix. So no longer just carbon, hydrogen, and oxygen. Okay. So proteins, all proteins are just long strings of amino acids. So the average protein is somewhere between like 30 to 100 amino acids long, but some of them can get much, much longer, to 300 to 1,000 amino acids, but very few of them are smaller or significantly smaller than that. And proteins are the main tool of the cell for essentially everything that it's going to do. Right? As I mentioned earlier, your DNA, your genetic information is all acting as instructions for your cells to make proteins. And then those proteins go and do that work, whatever was coded for in that DNA. And so these proteins, again, are strings of amino acids. So the monomer here, the building block that makes up proteins, is a single amino acid bonded to another one. Each amino acid has a central carbon with the same 3 groups listed here. And then that 4th bond left on the carbon is what changes between each amino acid. So every amino acid has a carboxyl group bonded to that central carbon, has a lone hydrogen bonded to that central carbon, an amino group bonded to that central carbon. But then the 4th bond here is this R group or variable group, which is going to be different for each of the 20 amino acids that living things utilize. And so just like with each of the organic molecules we've been talking about, dehydration synthesis would be used to form a bond, and hydrolysis would be used to break it. But we do have a special term for the bonds that hold amino acids together. And this is referred to as a peptide bond. Okay. Strings of amino acids are also referred to as polypeptides, or early immature versions of proteins might be referred to as a peptide or polypeptide. Okay. And that is in reference to the peptide bond being formed between the carbon in that carboxyl group and the nitrogen from the amino group. Okay. And ribosomes are what create this bond. Okay. So ribosomes are the organelle that make all of your proteins. So they're going to take one amino acid at a time, create a peptide bond between them, and just continue that process, building this chain longer and longer until we get a long linear chain of amino acids. So this is our primary structure for proteins. Proteins have 4 levels of structure that we're going to talk about here. So the primary structure is how it would be leaving the ribosome. So after the RNA has been translated and the ribosome has attached each of the amino acids that were coded for in that RNA, we now have this long chain of amino acids. But the protein is not functional in this form. So immediately after release, the amino acids in that linear chain are going to start forming those hydrogen bonds, those magnetic interactions with each other. to give them their secondary structure. And so secondary structure is either as a alpha helix or as a beta sheet. But again, it's just starting to give the protein some shape. Right, so still in this secondary structure, this protein is not yet mature and in the form it needs to be to perform its function, to perform what it was designed to do, but is now starting to get some shape. Again, these are hydrogen bonds, those magnetic interactions, creating our secondary structure. We then move into our tertiary structure. And so many proteins are done at this level, or they are functional at this level. So our tertiary structure is typically going to be formed from some other protein. Some other protein will be involved in helping bend that string of amino acids into the shape that it's going to have here in its tertiary structure. And the bonds holding this tertiary structure together are these very strong disulfide bonds. So two sulfur elements involved here, these disulfide bonds are very, very strong that hold amino acids together into this tertiary structure. And our last level of complexity here is our quaternary structure. So primary was first, secondary, second, tertiary, third, quaternary, our last form here. And so not all proteins need a quaternary structure, but they have this option, or some of them can. So a quaternary structure is essentially a complex that has multiple polypeptide chains together. So multiple of these proteins that were in their tertiary form have formed interactions to create this large complex of 4 of these hemoglobin proteins, creating the full mature hemoglobin protein itself. Okay, so these are actually 4 polypeptide chains that have now been bonded together. Another example would be keratin or collagen, those long fibrous proteins that make up the cytoskeleton. So primary structure, long chain of amino acids. This is what comes out of the ribosome. Secondary structure is those natural bonds that form from the charges that amino acids are carrying. Tertiary structure is a much stronger bond, giving it its 3-dimensional shape. And most proteins are active and mature in their tertiary form. And then quaternary structure has multiple polypeptides bonded together. And then there's all that together. All right. So like I said, proteins are really, really important as tools of the cell carrying out all of the instructions from the DNA. And one of the ways that they are very deterministic of a cell or of an individual is based on the enzymes that your DNA can code for. So enzymes are proteins that play a role in chemical reactions. Enzymes make chemical reactions happen faster and easier by essentially reducing the amount of energy needed for that chemical reaction to occur. So most enzymes are quite specific to what molecules and what reaction they play a role in. They each will have these active sites that have a particular shape and chemical composition that allow them to bond with very specific reactants or substrates here that essentially just brings them closer together and makes it much easier for a bond to be formed. And now we have our new product, right? So this was glucose and fructose. We brought them closer together, created a bond, and now we can release those as a single sucrose molecule. And again, enzymes do this by reducing activation energy. So every chemical reaction is going to require some sort of energy to get that reaction started. That is our activation energy. Enzymes just lower the amount of energy needed for that to occur. So without an enzyme, the amount of energy displayed on the y-axis here would be very high in order for this chemical reaction to occur. But with an enzyme, that energy need is much, much lower. And so this reaction can happen much faster and much more frequently. I'm really sorry, guys. I know this is a lot of information being packed into this, and it's been a long lecture so far. We've got 10 more minutes to get through just our last biomolecule here in ATP. So thank you for bearing with it. So again, our last biomolecule here, these are nucleic acids. Okay. More commonly, you'll see the term nucleotide to describe our individual nucleic acids. And these nucleotides are what make up DNA and RNA, so that genetic material. And so a nucleotide is made up of a ribose sugar with a phosphate group, as well as a nitrogenous base. And these nitrogenous bases are what are different between each individual nucleotide. And this is what is used to produce the code, the gene that is stored in your DNA or being carried in RNA, right? So we have 5 different nucleotides that make up DNA and RNA. Some of them are unique to only DNA or unique to only RNA. So we have our purines, which are adenine and guanine, A and G. We have our pyrimidines, cytosine and thymine for DNA, or cytosine and uracil for RNA. And again, we use these letters when notating them. We're going to do some review and work or practice with gene expression in the next chapter, where we'll come back to looking at those nucleotides more closely as being used for the genetic code. Essentially, the important thing here is that they have this nitrogenous base, which interacts with the nitrogenous base on the other line of DNA here. Right, we have 2 strands of DNA that form this helix, this double helix that you're typically familiar with when thinking about DNA. And so those nitrogenous bases in the center create bonds with each other. And they are specific, meaning that thymine can only ever bond with adenine. Adenine can only ever bond with thymine or uracil, which is the equivalent to thymine in RNA. And then same thing goes for the relationship between guanine and cytosine. Guanine can only ever bond with cytosine and vice versa. It's the main thing we'll cover there. Looking a bit about genes, protein synthesis. Okay, the last part here, the last major molecule that's going to be really important for this class is ATP. So ATP is the molecule that carries energy throughout the cell and is a much more accessible source of energy for all parts of the cell. So even though there is energy stored in molecules like carbohydrates, lipids, and proteins, it is much more difficult or requires much more specific enzymes to break apart those bonds as a source of energy, whereas ATP can much more easily break these phosphate bonds and release a high amount of energy for work to be done at any part of the cell. And so ATP is made up of a sugar or a couple sugars here with ribose and adenine. And then the T is referring to tri because there are 3 phosphate groups with each of those bonds between these phosphates holding a high amount of energy. And so ATP is constantly being cycled between 3 phosphate groups and 2 phosphate groups. So when it is providing energy, it loses one of its phosphates and goes down to ADP, so diphosphate. And then we can add a phosphate group back to it somewhere like the mitochondria or through respiration earlier on, like glycolysis. We can make a little bit of ATP by adding a phosphate group back. And that's essentially it here. Okay, so we're constantly breaking and then reforming that bond to cycle energy in our cells. Whew. That's always a long one. And next week, going over cell bio, again, is also going to feel like a really long one because it is reviewing, again, basically an entire biology course. Right? You would have learned this stuff in a biology course over a couple of weeks. You would have learned what we're going over next week over the entire quarter. So it would be a really good idea to go ahead and reflect on those major concepts or look ahead to the PowerPoint or read through the textbook before class because I'm going to be working at a similar pace, trying to fly through that PowerPoint, fly through that review as quick as possible so that we can move on to our actual new anatomy and physiology content. We do still have a few minutes here, so if you have any burning questions, comments, or concerns, please feel free to share.
The pace that we're going right now is more because it's just a review and it's not necessarily reflective of the rest of the class.
Yeah, yes. So there are still slide decks that are quite long, but as we move further through this course, I'm going to more and more focus on like major concepts from each chapter and expanding on or spending time on those major concepts, because those are the things that you're going to be applying in real life or in your career, or how you're going to use that info. And so I will be relying on you guys to spend time with some of the particulars and some of the smaller details from each chapter, so that class time can be a bit slower and direct and focused, right, on specific ideas. Whereas right now it's just, you know, a mile wide and inch deep, trying to make sure we touch all of it to show you like which stuff you need to remember from before. Any other questions, comments, or concerns here, guys? Okay, again, thank you for sticking through this with me. I hope you all have a great weekend, and I will see you on Monday for our first like real lab. So have a good weekend and see you then.
All right.
Thank you.